All right, our last balance is acid-base balance. So again, balance is just indicating that gains have to equal losses in order to keep us at homeostasis. And the level where we're looking to keep blood pH is what value? Now, if you said 7.4, you are correct, but we have a little bit of wiggle room in here. Most things aren't like a single point, and so really the body is happy as long as pH is between 7.35 and 7.45. I'm not a huge one for memorizing numbers. That's a good one to know, right? This comes up really commonly. Diabetic ketoacidosis is a major it's a major situation, and it's actually pretty common, right? A reason that people would be seeking medical care. So blood pH needs to stay within these boundaries. Now, what's the big deal? If pH drops below this, you're in trouble, right? Particularly that low pH, it starts interfering with the cell membrane, denaturing some of those transport proteins, the enzymes embedded in cell membranes stop functioning. And really below, it's around 6.9 is lethal, right? So this really is an important value. And so what we're going to be looking at in here is kind of where these acids are coming from and how we're able to balance them out. So we have multiple different classes of acids. Now, I will say this before I launch into it. This is the one that we're going to talk about the most, okay? So fixed acids, these are things like sulfuric acid and phosphoric acid, and they're really not ever in huge amounts. They come from trying to break down amino acids, the catabolism of amino acids, but they're pretty minimal involvement, right? So there are fixed acids. What it means is they don't leave solution. And so since they don't become gaseous like a volatile acid, it is the kidney that's going to have to step in and actually remove those from the body, right? But you don't really see those getting out of whack. These organic acids are going to be the byproducts or part of our metabolism. So these are ones you're super familiar with, things like lactic acid that we saw from anaerobic metabolism, and then ketone bodies. This is part of that diabetic ketoacidosis. The individual is trying to break down fats because their cells aren't utilizing glucose. Now, the thing with both lactic acid and these ketone bodies is they actually still have a lot of energy in them, right? They were both meant to be fuels, but in anaerobic respiration, recall, all we did was glycolysis. We didn't continue to the point where we got all those, 36 or whatever ATP. So the energy is still in there. And so typically, these two, or any metabolic acid really, ends up being metabolized pretty quickly. In this case, the liver is going to take those molecules and continue the breakdown to get the energy out of them. So again, these don't end up playing a big role, right? They don't accumulate in the body. So the one we'll talk about the most then are these volatile acids. Volatile just means that this can become gaseous, right? So we know that these can leave the body through the lungs. Do you see it? It's your equation. It's your favorite equation. Here we are again. So really, what we're looking at, right, are our hydrogens here are the part that would be making the body more acidic. And if you recall, right, if our equation moves this way, right, we are going to exhale that CO2, effectively removing hydrogen, right? And so that's the way this volatile acid is going to be removed from the body at the lungs, right? So 3 different classes of acids. Okay, so then we need to think about, well, how do we keep these acids in check? And here's the big picture, right? Basically, our first option are to use buffers, something that's going to kind of temporarily keep our pH from swinging too far one way or the other. After that, we really do need the lung or the kidney to get involved and really remove the acid. But let's start by looking at these buffers, okay? Now, there's a few different ones. Let's start here in the middle with these protein buffers. They're pointing out the difference here. Some of these really only work in the intracellular fluid, like the phosphate buffer system works in the ICF and also actually in the urine. Protein buffers work in both, and then carbonic acid is all extracellular fluid. But the important thing here, proteins. So proteins are able to act as really good buffers. Sometimes we see, like you'll recognize this from the respiratory system, right? Hemoglobin actually acting as a buffer when we got the dissociation of carbonic acid into bicarbonate and hydrogen. That hydrogen, recall, attached to hemoglobin so that we wouldn't see a huge swing in the pH of the body, of the plasma. Okay. All proteins are able to act as buffers because they're made up of amino acids. So if we look at an amino acid here, right? Wow, if it's been a while since you had Gen Bio, right? You have a central carbon that has a hydrogen and an R group. This R group is variable. Each of your 20 different amino acids has a slightly different configuration here, right? But what amino acids have in common is they have what's called the amine group. And this is the group that contains nitrogen. And then they have the acid group, because you see that hydrogen kind of hanging off the end. Remember, an acid is willing to donate hydrogen, and so that's true here. Our nitrogen is an interesting element because it can actually make an extra bond, and so it's willing to take on a third hydrogen. We call a molecule that's willing to accept hydrogen a base. Good. So in amino acids, one end acts as an acid and one end acts as a base. That makes them an amazing buffer, right? They can buffer changes in pH of either direction. And that's actually what they're showing here, right? With a higher pH, that hydrogen falls off. It's donated to correct the situation. If the pH is becoming more acidic, there's lots of hydrogen, that amine group is willing to pick it up and carry it around for a while, right? So proteins make excellent buffers, right? That's part of the reason why we have so many different plasma proteins floating around, right? Part of it is osmotic attraction, and then part of it is also pH buffering. Okay, the most important buffer system that we have is this carbonic acid bicarbonate buffering system, and this is exactly the one that we've been seeing all along. Let's just write it out one more time for the fun of it, right? That CO2 and water can join together to form carbonic acid, right? Remember, this is a reversible reaction. It can go either way. Carbonic acid. If it's an acid, that means it's willing to donate a hydrogen, is correct, right? And so that hydrogen and bicarbonate ion, again, a reversible reaction. So really, when we have carbonic acid, it's a weak acid. It is willing to donate a hydrogen. And when we talk about a buffer system, right, a lot of times this is the example of what we're talking about. A weak acid and the weak base that results when it dissociates, when it falls apart. So bicarbonate actually then acts as a base, right? It's willing to accept hydrogen and move the equation back this way, right? So those are our three most important buffering systems. Now there are a couple others, so they kind of fall in lines with like this phosphate buffer system. Actually, let me show you. right? So this is actually showing you that phosphate buffering system. There's two different versions of phosphate. One's a monohydrogen, one's a dihydrogen, right? So this is willing to take that hydrogen, right, and buffer it. And then this is the ammonia buffer system, right, where ammonia is willing to accept this hydrogen and become ammonium, right? That's showing you that this takes place in the urine. We'll come back to that in just a second. But those are buffer systems. Whoa, way too far. Those are buffer systems. I just went the wrong way as well. Okay, so when we're trying to maintain blood pH, our first stop is with the buffers, but again, this is a temporary fix. This is a Band-Aid on the situation. We can tie up extra hydrogen ions or donate them, but eventually we need the lung or the kidney to fix the situation. So here's what's going on at the lung. And again, you guys are super familiar with this. I like they've got different colors to show you which way the reaction is running. Now, this carbonic acid bicarbonate buffering system really is really good. The limitation that we face is if we ran into a situation where we're running out of bicarbonate, then we could no longer pick up those hydrogen ions, right, that are being made in a variety of different places. However, our body does have a reserve system. So sodium bicarbonate, i.e. baking soda, sodium bicarbonate is pretty prevalent in the body, and it can dissociate into sodium by itself and bicarbonate, and that bicarbonate then is donated back into this buffer system, and again, we can exhale to get rid of this CO2, right? And so remember those fixed acids and organic acids, they don't volatilize, right? They don't become gaseous altogether, but when their hydrogens get put into the solution, as long as we have that bicarbonate, we can also use that to carry the hydrogen away at the lungs, which is pretty clever. Okay. In the kidney, we also, one other thing, maybe two other things to keep in mind. The other limitation probably of this is that this system can't buffer when there's more changes in CO2, right? So it buffers, like this example, any of those acids that are added to the body, we get rid of. But say you're having a problem with your respiratory rate, you're hyperventilating or something, then if there's a low amount of CO2 because you're breathing too fast, well, that's the problem, right? So the lung is not going to fix that one. We're going to need buffers of the kidney or something like that to step in, right? So along those same lines, like if the lung isn't functioning properly, right? So maybe you have pneumonia or something's going on with that respiratory membrane, that can really inhibit the ability of this buffer system to work properly. Okay. At the kidney, I really think of the kidney as kind of helping the lung out. The lung can make these changes way faster. As long as the lung is healthy, it's going to do a pretty good job with most of these things. But the kidney is an excellent assistant. So this picture, 2713, is a little bit crazy, but the thing that you really want to focus on, right, again, they're showing reabsorption in blue, secretion in red. And so in this picture, we see a lot of reabsorption of things like bicarbonate and a lot of secretion of hydrogen, right? So that is one of the things we can do at the kidney is secrete hydrogen if blood pH is too low. As long as there are these buffers, the phosphates and the ammonias, in the urine to accept those hydrogens. If you run out of buffers in the urine, this diffusion no longer works, right? So you stop secreting hydrogens to the kidney. So you do need those buffers to be available, right? And so here they are, they're showing us actually some of those buffers we talked about earlier. Bicarbonate is present there, phosphate, and then ammonia, okay? On this side, notice you also can secrete bicarbonate and reabsorb hydrogen, probably less common, but the kidney really can work in both directions. Again, so we can variably, so we use the buffer, the lung, or the kidney. And really the question is, do we reabsorb hydrogen and secrete bicarbonate? Okay. You'd want to reabsorb hydrogen if your blood pH is getting too high, if you're becoming alkaline. Or do we want to secrete hydrogen and reabsorb bicarbonate like we see over here, right? So if blood pH drops too low, you can secrete it, okay? So all three of these systems, the buffers, the lung, and the kidney, are going to work together to help us maintain this very important parameter, blood pH.